Hello and welcome. Today's topic is our hemoglobin. It is the most important and most available metalloprotein present in our body. It has mostly histidine, lysine and isoleucine. Now this hemoglobin presents inside red blood cell RBC. Now what is red blood cell? I have talked about these things in my earlier cytology videos, you can check those. Now bone marrow mainly has hematopoietic stem cells. Among those stem cells we have specially proerythroblast. This proerythroblast gradually grows and converts into erythroblast. Now this erythroblast gradually converts into immature RBC or immature red blood cells. Up to this much, these cells contains nucleus. Inside the nucleus it contains nucleolus. So inside the nucleus it contains chromosome. So ultimately this hemoglobin synthesized from 11th and 16th chromosome. Now when the immature red blood cell becomes matured, at this process the nucleus and all its structure gets destroyed and the empty space is occupied by hemoglobin. Anyway, this entire process we'll talk about details in physiology chapter, not here. Anyway, our hemoglobin is actually quaternary structure of protein. This hemoglobin has 4 polypeptide chains, 2 alpha globin and 2 beta globin. This globin part is also known as subunit. Anyway, in some textbooks you can find 2 alpha subunit or 2 beta subunit. Anyway, now alpha globins synthesized from 16th chromosome and beta globins synthesized from 11th chromosome. One hemoglobin has 4 iron atoms, so it can bind with 4 oxygen atoms. Each red blood cell or RBC contains approximately 280 millions of hemoglobin molecules. Normal range of hemoglobin in our blood in adult male 13 to 16 gram per liter and in female 12 to 15 gram per liter. Look at those pictures, try to understand the structure of heme. Heme is actually iron porphyrin compound. So what is porphyrin ring? Wait. This porphyrin ring are actually complex compound with a tetrapyrrole structure. 4 pyrrole structure. Each pyrrole ring having the following structure. Look at the pyrrole ring above photo, okay? 4 pyrrole ring combines together or combines through methine or methylidine bridges to form one porphyrin structure, as you can see in this photo. And for extra knowledge point, the outer carbons of the 4 pyrrole ring which are not linked with the methylidine bridges are number 1 to 8, as you can see in this photo. Methylidine bridges are referred as alpha, beta, gamma, delta respectively. 2 hydrogen atoms in the amino group of pyrrole rings, second and fourth position, are replaced by ferrous iron, Fe2+, which occupy the center of the compound ring structure and establishes the linkage with the all 4 nitrogen of all the pyrrole rings. The iron, beside its linkage to 4 nitrogen of pyrrole rings, it also linked with internally fifth linkage to the nitrogen of the imidazole ring of histidine of the polypeptide chains. When hemoglobin is oxygenated, the water, the H2O, is displaced by oxygen. Formation of heme pockets. What is this? The hemoglobin molecule and its subunits contain mostly hydrophobic amino acids inside, internally, and hydrophilic amino acids are outside, at the surface. Each subunit, each globin contains one heme compound hidden inside hydrophobic subunit. The heme pockets of alpha subunit or alpha globin are sized just adequate for entry of one oxygen molecule, but the entry of oxygen molecule into the heme pockets of beta subunits are blocked by valine. You can say beta, delta, epsilon, gamma, zeta, whatever subunits are not allowed to contain oxygen due to the presence of valine. Almost 6.25 gram of hemoglobin are produced in our bone marrow every day, and the same amount get destroyed in the spleen every day. The structure of heme remains constant and recycled to form new hemoglobin in the bone marrow. Anyway, we'll talk about these things details in physics chapter, not here. See, our hemoglobin has high affinity to bind with carbon monoxide, then oxygen, and then carbon dioxide. How, why, we'll talk about later, not now. First of all, adult hemoglobin type 1, HbA1, 90% in our body with 2 alpha and 2 beta globins. Adult hemoglobin type 2 or HbA2, 3% in our body with 2 alpha and 2 delta globins. Then HbA1c, which is almost 6% in our body. When excessive carbohydrates in our blood attaches with the hemoglobin, it converts into glycosylated adult hemoglobin type 1. Anyway, this HbA1c we'll talk about later in disease chapter in details, not now. Then embryonic hemoglobin or HbE presents only in embryo stage with 2 alpha and 2 epsilon globins. Fetal hemoglobin less than 2% in our body with 2 alpha and 2 gamma globins. So about this embryo and fetus, I talked about details in my earlier embryology videos. You can check those. And lastly, we have adult hemoglobin type 3 presents only in old RBC with the 2 alpha and 2 zeta units. RBC lives around 120 days, I mean almost 4 months in bloodstream. So this type of hemoglobin found after 100th days of RBC. Now as you can see, this beta globin can be easily replaced by gamma or delta or epsilon or zeta to make other variants of hemoglobin. Alpha globin has 141 amino acids, and rest beta or gamma or delta or zeta or epsilon globin has 146 amino acids. So any type of one hemoglobin has totally 574 amino acids because 2 alpha globin and 2 other globins. Okay, there are also other type of hemoglobin found in disease condition, which I'll talk about later, not now. Now the hemoglobin also presents in dopaminergic neurons of substantia nigra, astrocytes of cerebrum, hippocampus, oligodendrocyte, macrophages, mesangial cells, alveolar cells. About all these structures, I talked about these things in my earlier anatomy videos. You can check those. And now the function, hemoglobin is more important in oxygen binding and its transport. So this metalloprotein also acts as a carrier protein and delivery this oxygen to every tissue, every cells which is required for metabolism. And after metabolism, there is the production of carbon dioxide, which is also carried by this hemoglobin towards the lungs for exhalation. So about this process we'll talk about later. Here you can find another special term, 2,3-biphosphoglycerate or 2,3-BPG, produced in red blood cells by Rappaport-Leu-Behring shunt or RL cycle, which is necessary for stabilization of hemoglobin conformation at quaternary level by holding salt bridges, and it's important for understanding of high altitude sickness and the changes in takes place of adaptation at high altitude along with ODC, I mean oxygen dissociation curve. Now what are the salt bridges, what is high altitude sickness, and what is oxygen dissociation curve or ODC? We'll talk about later these things in physiology chapter, not here. So see you in my next video. Then bye.